Our next presentation is from Dr. Hanu Pappu, and his presentation title is Tomato Spotted Wilt Virus Host Interactions. What is the host telling us? And so we're going to move on to that presentation. Just a little bit of background while we get loaded up. Dr. Pappu is a professor at Washington State University. He received his PhD at the University of Alberta in Edmonton, Canada. And he does a lot of work with genomics and biotechnology related to RNA and DNA viruses and across a lot of different crops. So I think just because of time, I'll step away from the introduction and let him get started. Thanks a lot for being here today.
Thanks, Dr. Jordan. I hope you all get to hear me.
I think we can. I think you need to be in presentation mode, and then I think we'll be good to go.
All right. See presentation mode. You can see my screen.
Yes, we're good to go. Thanks.
All right. Good morning, everybody. First of all, I'd like to thank Dr. Jordan and the program committee for inviting me to talk about some of the ongoing research in my lab about tomato spotted wilt host interactions. As David mentioned, I'm located at Washington State University in Pullman, so the northwest corner of the country. Today, I'm going to give a very brief introduction to taspoviruses and TSWV molecular biology, and then talk about some of the work that we've been doing on how hosts react to taspovirus infection using TSWV as a model system, and TSWV and tomato in particular as a model system, and how it relates to peanut for the focus of this meeting. And then I will talk about the host interactions and how the host responds to TSWV infection. And I will conclude that with an ongoing collaborative project with one of the speakers in this symposium at UGA, Dr. Sudeep Bagh, about developing RNA-based vaccines for TSWV using tobacco as a model system and how it is now being extended to peanut. To begin with, I'd like to acknowledge a collaborative effort. Some of the work that I'm going to talk about was done by one of my PhD students, Dr. Christian Olaya. The slide on the left, at the end of his defense exam, the guy who looks very happy in a suit and looking like a gentleman is the former PhD student Christian, who initiated some of these work, looking at TSWV and host interactions at the molecular level. And some of the committee members and Dr. Anna Whitfield, who was on the committee, joined virtually. And another postdoctoral fellow, Dr. Ramesh, and then a research associate in my lab, Dr. Inzai. So most of the work that I'm going to talk about is done by these fantastic researchers. And I have a long-standing collaboration, and I'd like to acknowledge Dr. Nina Mittar, a professor at University of Queensland in Brisbane, and Stefan Winter. He's a scientist at DSMZ in Braunschweig, Germany, and their lab members, Paulo Margaria and Stephen Fletcher and Jonathan Peters. So most of the stuff that I'm going to talk about was done by these wonderful collaborators, and I really like them because they let me take the credit. So very quickly, as Albert gave a very nice introduction and historical account of TSWV, it was first discovered in 1915, reported in Australia, and then for many decades, diseases having these symptoms were considered to be caused by TSWV. So there's only one virus causing a lot of these diseases in so many different crops. And then a breakthrough, a shift in paradigm happened by scientists at NC State, led by Dr. Jim Moyer, who showed that no, there is one more virus, it's not just TSWV. So the second taspovirus described in the late 80s was impatiens necrotic spot virus. So that really changed our understanding of what are these taspoviruses and the diseases caused by these viruses in different host plants. And the new taspoviruses continue to be discovered, reported from different parts of the world. So far now we have more than 32 distinct taspoviruses reported from different parts of the world. And Asia seems to be the most a center, if you will, for diversity of these task-force viruses. More and more viruses are being reported both in Asia and South America. Most of you know that majority of plant viruses are RNA viruses, nearly 80% of them. And a few of them are very different kind of RNA genome organization, like task-force viruses, which I will talk about a little just in a couple of seconds. As Albert mentioned, task-force viruses belong to a much larger family called Bunyaviridae, and majority of those members of those viruses cause disease in animals and humans. So viruses in this task-force virus group are the only ones that infect plants. So their worldwide distribution, as Albert mentioned, TSW has a very wide host range, severe economic losses, and these viruses are transmitted by thrips. A uniqueness about these thrips and task-force virus interaction is that these viruses replicate in the insect vector as efficiently as in the plant host. So now we have a situation where these viruses have two very different hosts, a plant and an insect, and managing these viruses continue to be a challenge because of that, breaking the cycle between the plant and the insect vector. So why task-force viruses continue to have great interest? Because these new viruses are being reported continuously, and existing viruses are expanding their host range, and new thrips vectors are being reported as the ability to transmit these viruses. And more importantly, resistant breaking strains are showing up. So some of the varieties that are developed with resistance or tolerance are not resistant anymore because of the emergence of new resistant breaking strains. So there is this continuous research and need for research to come up with new management tactics to manage these viruses. So Albert showed some of these nice pictures, and the one in the middle taken by Dr. Culbreth is probably one of the most widely used image showing the effect of TSWV on peanut. And bottom left is a tobacco, how the leaf is affected because of TSW infection. And on the right is a tomato on the in that picture. Early infection leads to severe stunting. So there are cases where total yield loss because of TSW infection. Quickly, the genome organization is a very unique virus compared to typical RNA virus infecting plants. It's a spherical RNA virus particle, and it has a glycoprotein envelope. And inside this virus particle, the virus has 3 RNA segments. Based on the size, they are referred to as large, medium, and small. Like most plant viruses, the virus codes for a replicase gene, and the medium RNA codes for a structural protein called that forms the glycoprotein envelope, and also it codes for a movement protein. So the small RNA codes for a nucleocapsid protein, which is unique for this group of viruses, that this nucleoprotein tightly binds to each of the RNA molecules. And like most plant viruses, or all plant viruses, TSW also codes for a very important gene called silencing suppressor. So the function of this gene is that it suppresses the host defense. So it's one of the most critical genes that pretty much all plant viruses are known to code for. So my lab has been focusing on the silencing suppressor gene and the interactions between this viral gene and the host over the last few years. And I'm going to talk a little bit about some of the findings that we reported recently. So again, so basically 3 pieces of RNA. And another interesting feature of these viruses is that the large RNA is in a negative sense, and medium and small RNAs are ambisense organization. So as a result, the virus particle always carries a few molecules of RNA polymerase enzyme. So upon infection, these RNA polymerase molecules initiate the first round of transcription to produce a positive sense large RNA. from which the RNA polymerase is produced in the infected cell, and subsequently the various steps of for replicating the small and medium RNAs can take place. So this is a cartoon basically showing the same, but large RNA codes for a polymerase that has a role in replication, and a movement protein, a glycoprotein envelope, silencing suppressor, and a nucleocapsid protein. So when virus infects a host, what does host do? It responds because it is now a war between a host and a virus, because host wants to defend itself. A virus wants to overcome the host response and establish itself and replicate. So in this case, it has been shown long time ago that plants fight back by a mechanism called gene silencing or RNA interference. And what happens because of this host response leads to degradation of the incoming viral RNA. And that results in small RNAs. How small, you may ask? Typically between 17 nucleotides to 24 nucleotides. So now there are methods and techniques developed to quickly determine the profiles of these small RNAs, which are a result of host response to virus infection. So to overcome host response, plants, viruses acquired this important property, as I mentioned, a gene that codes for a silencing suppressor. So this protein has been shown to interfere with this RNA silencing machinery in the host at various stages, so that the host response is disabled, so virus replication can take place. Right? So this interplay between virus and the host during the infection process leads to other side effects, if you will, that I'm going to talk about in just briefly. So in case of TSWV, what we have been doing is we developed a TSWV tomato as a model system to see how the host is responding to TSWV infection. And most of the findings that I'm going to talk about is very relevant and applicable to other hosts such as peanut or pepper. The other two commercially important and highly susceptible crops are susceptible to TSWV. So in case of tomato, there is a resistant variety that has a gene on SW5. It confers a high degree of resistance to TSWV. And so in varieties that have this SW5 gene, the virus infection is limited to inoculated leaves. The virus does not become systemic, as in the top cartoon. So we chose two varieties, one with resistance gene and one without, and looked at the host response to virus infection, and also what is the effect of the host response on virus small RNAs and their biogenesis in a resistant and a susceptible variety. So in a resistant variety, the virus is confined to only the inoculated leaf or infected leaf, whereas in a typical susceptible variety, the virus spreads to the rest of the plant and becomes systemic. So we have a very nice system to study not only the virus movement, but also the host response in an inoculated leaf versus a newly emerging uninoculated leaf that started showing infection and symptoms. So work done by my former PhD student, Christian Olaya. He basically compared the small RNAs that are produced in response to host defense in resistant and susceptible cultivars. So essentially, two varieties were inoculated with TSWV under controlled conditions, and RNA was extracted, cDNA libraries were prepared, and then high throughput sequencing was used to sequence the entire cDNA library that from each of the resistant and susceptible varieties. And then we established a bioinformatics pipeline so that all the sequencing data from these inoculated plants from two different varieties, resistant and susceptible, we filtered out all the host sequences and only selected all the viral sequences, that too the small RNAs, which are produced as a result of degradation of the virus by host defense response. So some of the results, it turned out that the viral small RNAs, result of a host RNA silencing machinery, that these virus small RNA profiles are distinctly different between a resistant and a susceptible varieties in terms of the size of the small RNAs, 22 nucleotides versus 24, two major classes of small RNAs that are produced due to the host RNA-I mechanism. So there's a distinct differences between the resistant and susceptible variety. And down the road, we are hoping that this can be used as a biomarker for screening breeding material. What kind of a small interfering RNAs are being produced in a particular breeding line. And then we looked at the viral genome itself, and these viral small RNAs, what is the biogenesis? What is their origin in terms of a large, medium, and small RNAs of TSWV? And it turned out in the resistant variety, the large RNA was the target. Most of the small RNAs that we detected in a resistant variety, they originated from large RNA of the virus. Whereas in a susceptible variety, most of the degraded small RNAs of the virus came from the small RNA of the genome. So again, there's a very distinct difference which viral RNA is targeted in a resistant variety versus a susceptible variety. This gave us some insights into what steps in the RNA-I mechanism, the pathway that this silencing suppressor either is targeting in a resistant variety versus a susceptible variety. This gave us some ideas in terms of what are the potential regions in the silencing suppressor protein that is interacting with a particular host protein. So which I'll talk about just in a few minutes. So again, we looked at which particular gene within the TSWV genome is being targeted by the resistant variety, the RNA-I submissionary. So again, it turned out that in a resistant variety, the RNA polymerase is the preferential target for degradation. Whereas in a susceptible variety, more of small RNAs came from the NSS gene that codes for silencing suppressor. Again, it makes sense if we look at the relative virus levels in resistant versus susceptible varieties, that we found more of small RNAs from RNA polymerase gene. Again, this gave us some ideas about designing targets or selecting targets in the viral genome to come up with the constructs that would provide a very effective RNA interference. Sudeep Bagh has been working quite a bit on this, using some of this information. Again, it turned out that a large RNA is the preferential target in the resistant variety versus a small RNA in the susceptible variety. So quickly, I will go to the next step where one of my previous postdocs looked at what are the targets in the host site. So far I talked about what is happening to the viral genome in a resistant variety and a susceptible variety. Now we wanted to look at what is happening on the host side, what host pathways are affected because of TSWV infection, right? So again, some of the basics, what we did at the time was all these small RNA profiles, the information of TSWV, then we looked at the propensity, that is the ability of these small RNAs to bind to host gene transcripts and alter the tomato transcriptome, again using TSW tomato as a model system. So as I briefly mentioned, upon viral infection, host responds to infection and degrades the viral RNA into small RNAs. But an unintended effect was that these small RNAs then bind to host genes, silencing the host genes. As a result, we see what disease symptoms in the plant. So this is somewhat a very surprising and unintended phenomenon that was described a few years ago, that these degraded small RNAs are not just disappearing slowly, but they do have a very important biological function and effect on the host genes. Right? So Ramesh then looked at some of these TSW small RNAs and how they are impacting the host transcriptome. So by using a suite of bioinformatics software, he identified quite a few targets in the tomato genome. And it turned out that many of them are involved in some of the very important biochemical pathways, including resistance response. And some of the viral small RNAs are targeting transcription factors in the tomato genome. And many of the hormone pathways, again, upsetting the host metabolism, leading to what we see as symptoms. So now there's an increasing body of information about what is the basic mechanism of symptoms that we see as a result of virus infection. These virus small RNAs are binding to some of the host genes, modulating their expression, affecting the downstream pathways, and leading to what we see as symptoms in the field. So now there are other ways to block this interference between the small RNAs and the host genes, leading to resistance phenotype. So there's a lot of potential applications of this information. So I don't want to go through all the list of various pathways that are potentially affected by TSW infection. But we have quite a bit of information on this. And bottom line, it turned out that many of the transcripts in the tomato genome that are affected by TSW small RNAs happen to have a very important role in both the genes that are expressed to biotic or abiotic stress. Many of the resistance genes. Right? So there are very distinct differences between resistant and susceptible varieties in terms of the changes in the host transcriptome. Right? So very quickly, some of the take-home message that we have a better idea about how TSW small RNAs are targeting what kind of a pathways in the host genome, either regulating, upregulating, or downregulating, depending on the genotype, depending on the genetic background, resistant versus susceptible varieties. And the last part of my talk, most recent project that we just recently published, maybe it came out a couple of months ago, is to look at specifically what are the host interacting partners that are involved in interaction with the TSWV NSS. Again, as I mentioned, NSS is a silencing suppressor. It interferes with the RNA silencing mechanism, disabling the host defense, enabling the virus establishment. Right? So it's a very critical viral gene for plant viruses in general. So we know quite a bit about NSS, but we don't know much about what are its targets in the host, because for a long time, we don't have a reverse genetic system, or what you call an infectious clone. So only recently, a Chinese group reported an infectious clone or reverse genetic system for TSWV. So Dr. Ying Zhai, a research associate in my lab, just recently published where came up with a very interesting approach because we don't have a reverse genetic system. So we've been using somewhat an indirect approach to look at TSWV host interactions. Right? So something that I already mentioned, it turned out that many years ago, it's been shown that the silencing suppressor is a virulence determinant of the TSWV. Resistance gene is TSW in peppers. And so it has the potential critical amino acids that are involved in this interaction have been mapped to a particular amino acid in the NSS protein. And as I mentioned, we don't know much about the protein interacting targets in host plants. So Ying used a very interesting approach where she picked up the NSS gene from TSWV, fused it to green fluorescent protein, developed a construct for agroinfiltration, and then subsequently she used affinity purification and mass spectroscopy to look at what are the host proteins that are binding to the viral NSS protein. And she came up with a very long list of proteins that are potentially interacting with the viral NSS. And some of the important proteins that were selected were a calmodulin protein, or CAM. And also it turned out that the heat shock proteins are also interacting preferentially with viral protein. So subsequent bioinformatics analysis, and we looked for homologs from what we found in Benthamiana, homologs in tobacco, in tomato, and Arabidopsis. And in all of those, across all those plants, these are the major ones that seem to be interacting with the NSS protein. These are heat shock proteins and CAM and MAPK. Right? And then she did a protein interacting network to see what are the major nodes of host proteins that preferentially or potentially interacting with the viral silencing suppressor protein of TSWE. And it turned out, again, CAM protein is a major one, seems to be interacting with NSS. And this is all based on bioinformatic analysis and mass spectroscopy. So then she verified this interaction between a viral NSS protein and the host CAM protein using Ease2 hybrid system. So most of you probably are familiar with the Ease2 hybrid system. It's a in vitro or in a heterologous system where you can study if two proteins are physically interacting. If there is an interaction, a particular yeast strain will grow on a selective medium. If the proteins are not interacting, you don't see any growth on the selective medium. So it's a very powerful technique to study protein-protein interactions. So she verified and confirmed that yes, the viral NSS protein and the CAM protein from tomato, or from Benthamiana, I'm sorry, are actually interacting. And the CAM3 has homologs in many other plant species. So conclusions, we identified several host proteins that are potentially interacting with the tomato spotted wilt silencing suppressor protein, and major nodes of these proteins are identified in protein-protein interaction networks. And some of these proteins, we are further looking into the host proteins to identify what are the specific motifs that are preferentially targeted by NSS protein. So I will skip, for the lack of time, the last part of the talk, but the future, we think that it's very exciting in looking at TSW and host interactions. Quite a bit of progress has been made in many labs around the world. We know more about the virus, but more and more it's now being known on the host side. Knowledge about the molecular mechanisms of TSW host interactions has a great potential for developing novel approaches for virus management. For example, genome editing methods like CRISPR, that now recently we established a platform for CRISPR looking at TSWE resistance, how to develop resistance to TSWE, modifying genome of TSWE. I'd like to thank Dr. Jordan again for the invitation, and funding for some of this research came from USDA and IFA Specialty Crop Research Institute and a host of other funding agencies. So I think I used up my time. I'm sorry. I'll stop here.
Thanks, Dr. Pappu, for the really informative presentation. We'll take one quick question, and I've got one here. Does degradation of the viral genome occur? Let's see, it skipped on me here. Does degradation of the viral genome occur in both resistant and susceptible varieties, resulting in small RNAs?
Very good question. Yes, it does. I went over very quickly because that study was published a couple of years ago, but yes, it does. And more effectively in resistant varieties than in susceptible varieties because of the resistance gene in the resistant variety. But smaller RNAs are produced, yeah, absolutely in both.
Okay, thanks a lot. And if there are more questions, we'll catch some of those at the end. Again, thanks for joining us today. I know you're out on the West Coast, so we appreciate you being here and being on the program with them. Thanks a lot.